Hello guys, welcome to our second part of food and digestion, the digestive system. This time we're gonna carry on. We're gonna do enzymes first, so digestive enzymes. So enzymes, I remember, are biological catalysts that help biological metabolical reactions in our bodies. In this case, digestive enzymes, they break down substances. The three main food substances, carbohydrates, proteins, and lipids, are broken down by three classes of digestive enzymes. And conveniently, these enzymes are actually named after the substance they break down. So carbohydrates are digested by enzymes called carbohydrases. Proteins are digested by proteases. Lipids are digested by lipases. Easy, right? So carbohydrates, amylase is a carbohydrate. It is produced in the salivary gland and the pancreas. It breaks down starch into maltose. Maltase is secreted from the wall of small intestine. Its role is to break down maltose into glucose subunits for absorption. So this I don't want you to worry about that much, but the next ones I want you to draw them. This one is simply because it's a little bit complicated to draw, but I'd like you to see how amylase digests starch into maltose, and then maltose is digested to maltase. By maltase to 2 glucose subunits. Yes, carrying on. Proteases. So first one is pepsin. Pepsin breaks down proteins into peptides, which are smaller chains of amino acids, and it is secreted from the stomach wall. Trypsin does the same thing, breaks proteins into smaller peptides. Like peptides, like I said, they are smaller chains of amino acids. They are not individual amino acids. And it is secreted from the pancreas and works in the small intestine, specifically beginning of small intestine. Peptases, they break down peptides in individual amino acids for final absorption, and it is produced by the wall of small intestine. Okay. Here in this diagram you can see the stomach, and you can see pepsin, how it breaks down proteins into smaller peptides, and so on. So lipase is the last one. Lipase processed in the pancreas but acts in the small intestine. It breaks down lipids into glycerol and fatty acids, basically subunits of lipids. And you can see the same diagram. Breakdown of food into duodenum. So duodenum, from the mouth to the initial part of the small intestine, as you remember. And we're gonna go step by step how food progresses towards our gut. So when we eat, the digestion of food starts in the mouth. Saliva moistens the food, and amylase in it starts to break down starch, while teeth chop down food into smaller pieces. So this actually was called mechanical digestion, as you remember, chopping down food into smaller pieces, and the digestion by the amylase is chemical digestion. So the chewed ball of food goes down to the stomach, where it is held for several hours. The HCl, hydrochloric acid, an enzyme pepsin, is released here. HCl, hydrochloric acid, has two functions. First, it kills all the microenzymes ingested with food, preventing us from getting food poisoning or ill, and activates enzyme pepsin, which has an optimum pH of 2. Differently from most enzymes, pepsin actually has an optimum pH of 2, which is really low. As other enzymes, they have an optimum pH around 7, which is natural. After the stomach, semi-digested food passes down to the first part of the small intestine. It is where several other enzymes are added to the food. So up till now, there is no absorption. Okay, this is still digestion going on. Still breaking down going on. Sorry. Additionally to the enzymes, a digestive juice called bile is released from the liver, specifically from the gallbladder. So bile is stored in the gallbladder. The bile carries out a type of mechanical digestion on lipids by creating an emulsion of tiny droplets from larger pieces of lipids, increasing the surface area greatly for lipases to work much faster. So essentially, what it does the same thing as our teeth. It chops down the lipids, specifically lipids, into smaller pieces, increasing the surface area, making it easier for enzymes to work on. Sometimes people will have problems with their gallbladder. They will have a cyst in there, or a tumor, or a blocked bile duct. So what will they do? The doctors surgically remove the gallbladder. But from now on, people that had the surgery, had the gallbladder removed, they should eat a diet which is poor in lipids. So they cannot eat a lot of lipids because they won't be able to digest them very well. So bile is essential for the digestion of lipids. The pancreatic enzymes and the bile has a secondary function of neutralizing the pH dropped to 2 by stomach HCl. So as you know, most enzymes work around 7 pH optimum. So what bile does, it actually drops the pH down as well. So that helps. Basically, it doesn't. This acid does not harm your innards in the ileum, your small intestine. After the food is fully digested, the smallest units of carbohydrates, proteins, and lipids are absorbed to the blood in the ileum. Ileum is the second part of the small intestine, the rest part, not the initial, but the whole small intestine. So what happens here is absorption. So absorption of nutrients in ileum. The ileum is specifically adapted to absorb digested food. It has a really high surface area to aid absorption of nutrients. This surface area is achieved by three things. The length of the small intestine, which is around 8-9 meters, about 8 meters, 7 meters long for an average adult human. And you have inner surface with folds. Addition to that, you have a macroscopic projection called villi, and in addition to that, with which I'm not gonna go in detail about a lot, but those microscopic projections also has microvilli. So first you have a really long tube which has folds, further increases the surface area. So it doubles, maybe triples, quadruples the surface area. Then you have your villi, microscopic projections, as you can see in this diagram. And on that villi, you actually have cellular, really tiny hair-like structures. So microvilli we call them. So you have an amazingly high surface area. What this does, it helps to the absorption. Okay, the nutrients are they're really they absorb really fast as they go down your small intestine. So carrying on, singular of villi is a villus. Each villus are small hair-like structures about 1 to 2 millimeters long. Inside each there is good blood supply, but a lot of capillaries which actually absorb and carry nutrients around the body. So here in the diagram, which I want you to draw into your notebooks, you have the small intestine, you have the folds, and you have the villi, and you have a single villus. As you can see, you can actually see the capillaries and the veins, arteries supporting them, and you can see actually lacteal. What lacteal is, I have a description in the following slides. But essentially, what it is, it carries out a different fluid. Other than it carries a plasma, which is blood without red blood cells, but you have white blood cells. And what's function it has in the villi is that it carries out, it transports the product of lipid digestion. So it carries fatty acids and glycerol, but eventually it actually meets with blood. So third part, elimination of waste, digestion, the large intestine, which is colon. As gut contents reach the large intestine, the colon, there is very little food and water left, and most of it is just cellulose, which is fiber, as you know, or other undigested material. This can be dead cells, dead bacteria cells, dead human cells, lining of the gut, cells in the lining of the gut. If you're a different animal, maybe bones, hair, and etc. We can divide the large intestine into three parts. First one is the colon, the largest part, the whole from beginning till the rectum. It is where the most of the remaining water is absorbed and the feces are formed. So water is absorbed, then you have compost, which is actually feces. The rectum, it is where the feces are stored, and the anus is where the feces are discarded out of the body. So that's essentially what the route food takes when we eat. A few extra attempts, maybe you see them in the diagrams previously. So sphincter muscles, these are muscles located at the end of your stomach, your anus, and the bladder. They control passage through these organs by muscular contractions. But they contract, they close down the passage, they relax, they release the contents. Digestion is the removal of waste and undigested parts of the food eaten. So it's not actually, not to be confused with excretion. Excretion, not to be confused with digestion. Excretion is the removal of cellular metabolic waste, such as CO2 and urine. It is carried out by our kidneys. Okay, there's a little typo here. So it should be our kidneys. So indigestion is the formula of feces from undigested food parts. So it's not actually something that your metabolism forms. On the other hand, excretion is metabolic waste such as CO2 and urine. Microvilli, structure similar to villi, located on each villus, increasing the surface area even more. So the stuff I told you about. Villi, microvilli is villi on villi, in a sense. Carrying on, lacteal structure similar to capillaries inside the villi of small intestine. However, these vessels carry products of lipid digestion, like I mentioned, glycerol and fatty acids. Yes, we came to the end of digestion and enzymes, digestion and digestive system. So this is all we're gonna do about this topic. I will send you a homework as well, and no video today, and I included a checklist here, so let's read it. Pay attention to the lecture and my sentences. Write down the notes on the slides to your notebooks. Have a look at the videos I recommend to you. Last time the video was sufficient, so essentially the video mentioned all the things we talked about today, so no video today. Next time. And do your homework and revise. So I will send you your homeworks as well today. And yeah, talk to you again soon. Thanks.